The overall goal of this procedure is to use a genetically encoded FRET bile acid sensor to easily visualize bile acid transport and dynamics with high spatial and temporal resolution.
This sensor can answer key metabolic questions, such as the relation of bile acid dynamics to glucose, lipid, and energy metabolism.
The main advantage of this technique is that bile acid transport can now be directly monitored in single living cells without the need for labeling the bile acids.
This method can provide insight into cellular bile acid transport and dynamics. It can also be used to visualize FXR activation in single living cells or to combine imaging of protein localization and bile acid transport.
The genetically encoded bile acid sensor, henceforth the sensor, consists of a donor cerulean fluorophore and an acceptor citrine fluorophore. The two fluorophores are linked by the ligand binding domain of the FXR nuclear bile acid receptor fused to an FXR cofactor peptide. Two engineered amino acid changes in the two fluorophores promote intramolecular interactions between the cerulean and the citrine. Two different constructs carrying the sensor are utilized. In one construct, the sensor contains a C-terminal nuclear localization signal and therefore accumulates in the nucleus. In the other construct, the sensor lacks any targeting sequence and thus localizes in the cytoplasm. The following video protocol focuses on how to image the sensor in living cells.